Throughout this class so far, we've talked a lot about antimicrobials, intrinsic resistance, and some organism-specific issues with antimicrobial resistance. Today, we're going to try and sum some of this up into a discussion of antimicrobial stewardship, how to use drugs appropriately, but perhaps more importantly, I hope to give you some of the tools to be able to think critically about how you'll prescribe these drugs. We're going to start off today with some figures that I showed way back at the beginning of the course, just to contextualize the problem. Over the last couple of years, COVID has certainly been the infectious disease that's really top of everyone's mind. But the slowish burn of antimicrobial resistance and kind of this ongoing pandemic that people really aren't talking about may end up being more damaging over the long run. The issue of antimicrobial resistance has been the topic of a number of large, high-profile international reports over the last few years. This particular report was famous for some of its infographics, including this one. What the researchers did was project it out into the future and said, what is the landscape of AMR going to look like in 2050 if we don't sort of change course? And very famously, what they found was that by 2050, there would be 10 million deaths a year directly attributable to antimicrobial resistance, which is more than are currently dying of cancer. So it's a very, very big problem, or it will become a very, very big problem. More recently, this study published in The Lancet found that resistance is already a huge problem, and we are well on our way to at least meeting, if not surpassing, those 2050 projections. So this study found that there were 1.27 million deaths directly attributable to resistance over the study period. So we're already reaching a high human cost associated with resistance. Of course, resistance is something that isn't sort of uniformly problematic across the prokaryotic world. There are some organisms that are a bigger issue than others. Some of the most pressing ones are things like our carbapenem-resistant Enterobacteriales, ESBLs, multi-drug-resistant Pseudomonas, MRSA, etc., etc. This is a list that's put together by the US Centers for Disease Control and was very famously published in 2019. So that's where we are today, but how did we really get here? So antimicrobials have been around since the 1940s, and even back at the time that these drugs were first introduced and new compounds were being discovered, it was recognized that resistance was an issue. So this is a quote from Dr. Mary Barber, an early researcher in antimicrobial resistance. She described some of the first reports of penicillin resistance among Staph aureus. And what she very astutely noted in this 1955 paper is that it's a neck and neck race in which many of us tend to underestimate the opponent. Staphylococci will not be defeated by the haphazard use of each new antibiotic. As new antibacterial agents are discovered, let us use them with discrimination. So very, very insightful and really hints at the needs for stewardship, even at the beginning. I've put a link above to a really interesting experiment that was done at Harvard University, where they exposed bacteria to antimicrobials at increasing concentrations over a period of time, and were able to demonstrate tolerance among these organisms sort of within the study period. Now, this doesn't exactly equate to antimicrobial resistance, but I think it's a good indicator of just how quickly bacteria can adapt. If we look back over the history of antimicrobial drug development, it's usually only a few years between when the drug was first introduced and when resistance was first identified. This is a table from the US Centers for Disease Control, and what you can see here, penicillin was released in 1941, and it was in 1942 when penicillin-resistant Staph aureus was first found. So it happens relatively quickly. And I think the take-home message here is that drug usage selects for resistance. It's almost inevitable. There's a group of organisms which we know are particularly problematic, and those are the escape pathogens. ESCAPE is an acronym, stands for Enterococcus faecium, Staph aureus, Klebsiella pneumoniae, Acinetobacter baumannii, Pseudomonas aeruginosa, and Enterobacter species. Resistance in some isolates of these organisms is such that there are no treatment options available. So really, really big threats. If you were to identify one of these escape organisms, be prepared for a nasty susceptibility profile. The Canadian Antimicrobial Resistance Surveillance System report, most recent data available is the 2022 report, which goes up to the year 2020, describes what the landscape of resistance is like here in Canada. And importantly, we can see from this summary table that many of the problematic organisms are trending up. So they are becoming more common. MRSA, VRE, carbapenem-resistant Enterobacteriales, Neisseria gonorrhea, strep pneumo, and salmonella infections. So the problem of resistance is getting bigger. If we zero in on just one of these groups of pathogens, our carbapenemase-producing Enterobacteriales, what you can see here is this rise in cases of healthcare-associated carbapenemase producers. Most commonly in Canada, we see KPC, NDM, and the OXA48 types. So these are the specific groups of enzymes which are most prevalent. And what you might notice on this figure is that we have this drop off from 2019 to 2020. What the authors of this report hypothesized is that this was really due to COVID travel restrictions. So in Canada, we are not yet a country of high endemicity for carbapenemase producers. And so many of these infections have some sort of travel association with them. And I suspect that as we go into the future, we're going to see these numbers continue to trend up as travel resumed sort of mid-pandemic. In this figure here, you can see the frequency of each type of carbapenemase over time. So our KPC type enzymes are sort of gradually decreasing, our NDMs are maybe going up a little bit or staying stable, and our OXA48s are becoming more common. So resistance is clearly an issue, but because resistance follows use, it's also important to look at how antimicrobials are used in Canada. This is some more data from the CARS report, and what they've done is compared antimicrobial use in humans to antimicrobial use in people in European countries, where we have really, really good levels of data. Here, what you can see is that Canada is actually starting to fare quite well. We're one of the lower usage countries. This breaks it down by community sector and healthcare sector. So receiving drugs in hospitals is epidemiologically perhaps different than in the community. Very interestingly, they found that between 2017 and 2021, prescriptions in the community decreased 30%, so perhaps reflecting increased stewardship initiatives. However, they also found that among hospital antimicrobial prescriptions, 77.5% of them were appropriate. And that sounds pretty good. But what this also indicates is that 22.5% of them were not appropriate across the country, which means that we have room for continued improvement. And there is a greater need for more stewardship initiatives in the country. In animals, it's really challenging to quantify antimicrobial use in a meaningful way. Trying to compare how drugs are used in chickens to cattle is really difficult. These animals are really different sizes, and so simply comparing the mass of antimicrobials used doesn't really convey the whole story. On the right, what you can see is a breakdown of multiple drug classes by year, so 2018, 19, and 20. Our tetracyclines, macrolides, and penicillins were the most commonly used drugs. And I think maybe we can appreciate there are some trends happening, a decreasing trend of macrolides, perhaps decreasing with sulfonamides. On the left, what you can see is quantity of medically important antimicrobials. So these are the ones which are of interest to human medicine by species. And this is the quantity of drug adjusted for both the population of animals and also their weight. So we can see again over those same 3 years, 2018 through 2020, perhaps a decrease in swine and poultry, and maybe a slight increase in cattle and our companion animals. I think it's really important to point out that this data is presented at a very, very high level and over a relatively short period of time. So the impacts of antimicrobial stewardship initiatives on specific drug families are perhaps not captured very well by this data, and maybe doesn't reflect the true state of how drugs are being used in Canada. In this figure, what you can see is the massive drugs used in animals in Canada as compared to those same European countries. So the Canadian data is in red here, while each European country is one of these histograms. And here, I think what you can appreciate is that compared to Europe, we're maybe on the higher end when considering how much massive antimicrobials are used. So again, we have room to improve. Stewardship initiatives have value. In companion animals, there have been some really interesting studies over the last few years. So these are some tables from this paper out of the UK. It's getting a little bit old now, but they did such an amazing job of capturing drug use in a large number of practices. So they collected data from 216 practices with a patient population of over 400,000 dogs and over 200,000 cats. And when they looked at which drugs were used most commonly, they found that overall, 43.6% of all antimicrobials used were beta-lactams. So some of our penicillin type drugs, cephalosporins, etc. When it was broken down by species, so looking at dogs versus cats and looking at the specific beta-lactams, a really interesting trend appeared. For dogs, amoxicillin with clavulanic acid was the single most commonly used drug, while in cats, it was a third-generation cephalosporin. And I suspect that this can be attributed to the introduction of cefovicin, which is a product marketed as a long-acting antimicrobial with the brand name Convenia. More recently, a similar study was done in Australia. These investigators looked at the records from more than 4.4 million consultations of dogs and cats, including the medical records from nearly 200,000 cats and over 500,000 dogs. There were 176,000 antimicrobial prescriptions, so really, really big data. They found a very similar trend. So again, cefovicin, the #1 most commonly prescribed drug in cats. This accounted for 32% of all antimicrobial prescriptions in cats. While in dogs, amoxicillin plus clavulanic acid was the most common, accounting for just over a third of all prescriptions. So I think at this point, I've highlighted that resistance is a problem. We know that we're kind of heading in the wrong direction, but what's the value of stewardship? Is this really all sort of doom and gloom? And with this figure, I hope I can convince you that it is not. There is reason for hope, and there's reason to try and change and improve how we are prescribing these products. So this is, again, a table from the CARS report. And what you can see here is the frequency of ceftriaxone resistance, so third-generation cephalosporin, in Salmonella, in both chickens and people, as well as how commonly these drugs were used. So back in the early 2000s, it was recognized that we had relatively high rates of third-generation cephalosporin resistance in bacteria isolated from chickens, and high rates of ceftiofur use. So this is a veterinary labeled third-generation cephalosporin. This led to some changes in how these drugs were used and a voluntary withdrawal from the poultry industry, which subsequently led to this drop in the frequency of resistance in both poultry and human isolates. More recently, policy changes were introduced that affected how these drugs could be prescribed and actually removed ceftiofur from use in the poultry industry. So in 2013, 32% of flocks were using ceftiofur. In 2014, only 6%, and following that, the drug was not used at all. What you can see here on this chart is that the rates of third-generation cephalosporin resistance really fell off, whether that's chicken at slaughter in yellow, retail chicken products in blue, or human isolates in this sort of dark gray color. So stewardship initiatives and changing how we prescribe antimicrobials can have a real-world effect, not only on the frequency of resistance in surveillance samples, but also in those isolates which cause infections. Before we get into a more detailed discussion of stewardship, I think it's important to just briefly review how antimicrobials work. If you need more of a refresher, I'll refer you back to our antimicrobials lecture number 5. I'll put a link above. But basically, these drugs work by attacking physiological processes or structures which are unique to bacteria. So the cell wall, cell membrane, the machinery responsible for nucleic acid synthesis, metabolism, and organization, and then protein synthesis, all structures which are unique to prokaryotes. Bacteria have a similarly broad spectrum of resistance mechanisms. They can stop our drugs from working through a number of strategies, either decreased permeability, so they prevent entry of the drugs, active efflux, pumping it out, enzymatic degradation or alteration of the drug, which allows them to destroy or inactivate our antimicrobials, modifying the target, so kind of putting on a mask and disguising, using alternate metabolic pathways, so accomplishing the same physiological goals through a pathway that is independent of the antibiotic, or simple resistance by absence where they lack the drug target. It's important to know that all of these strategies can be either intrinsically expressed, so they can be normal for an organism, and this isn't what we're talking about when we're discussing the emergence of resistance, or after gaining genetic competence. So these are the superbugs. They have something that differentiates them from the wild type. There's a few different methods by which we can determine susceptibility. Some phenotypic susceptibility tests, which can be either categorical or quantitative. We can do molecular assays to look for the resistance gene, or other phenotypic tests to look for the gene products. I'd like to also reemphasize that resistance has a very specific meaning. Calling an organism resistant is designed to predict clinical outcomes. So a resistant organism is not likely to be treated successfully when you use a drug in the species it's intended for an infection at the site it's intended for. So we assume achievable drug concentrations at the target tissue based on the dosing regimen on the label, and of course, the target pathogen. On the flip side, if an organism is susceptible, then based on these same assumptions, we can say that we are likely to have clinical success. I've already alluded to this, but we can break resistance down into intrinsic and acquired. Intrinsic resistance is constitutive for an organism, while acquired resistance is not inherent. It's a decrease in susceptibility compared to the wild type. I think it's important to understand the differentiation between intrinsic and acquired resistance. It can give you sort of a bit of a grasp of what all those R's really mean on your susceptibility profile. Ideally, labs should not be reporting intrinsic resistance, but I think it can be really helpful also for selecting empiric therapy. One of the perhaps most intuitive examples of intrinsic resistance is that mycoplasma are intrinsically resistant to penicillin and actually all beta-lactams. They lack a peptidoglycan-based cell wall, and therefore they don't have the drug target. On this slide, you can see a link to EUCAST, the European Committee on Antimicrobial Susceptibility Testing, where they have great freely available resources on intrinsic resistance. And I've also put a link to a video above that I made where I kind of go through intrinsic resistance in a bit more detail to help explain this perhaps somewhat difficult to understand concept. In the next few slides, I wanted to go through some of the commonly recognized intrinsic resistance. So this is data that all comes from EUCAST, and as of producing this video in late 2023, it's the most up-to-date information available. Here we have our expected resistance phenotype, so kind of our intrinsic resistance for Enterobacteriales and Aeromonas. Up at the top here, I've highlighted the resistance that's common across the entire group. So all Enterobacteriales and Aeromonas are resistant to benzylpenicillin, glycopeptides, lipoglycopeptides, fusidic acid, most macrolides, lincosamide, streptogramin, rifampin, and the oxazolidinones. So these drugs will never work for any of our Enterobacteriales. Below in the table, you can see the species-specific information. So for instance, our Enterobacter cloacae complex is also intrinsically resistant to ampicillin and amoxicillin, or potentiated penicillins, as well as a number of cephalosporins and cefoxitin. This is just the remainder of the same table that I've included for completeness. One group of organisms that I think is worth remembering are the spice organisms. So Serratia, Providentia, indole-positive Proteae, which is Proteus vulgaris and Morganella, Citrobacter, and Enterobacter. These are all within the Enterobacteriales family, and they're worth remembering because they intrinsically produce AMP-C type beta-lactamases. The production of these enzymes can become de-repressed, so they can make more of it when they're exposed to an antibiotic. And The consequence of this is that they are resistant to many of our beta-lactams, including the third generation cephalosporins. So in a veterinary context, I would recommend against using any of the beta-lactams for treating the spice organisms. In this table here, you can see intrinsic resistance in our non-fermenters. Again, those intrinsic resistance phenotypes, which are common across the whole group, are listed on the top. And I just want to bring your attention to Pseudomonas aeruginosa, which is a very commonly encountered non-fermenter. Again, resistant to our penicillins, potentiated penicillins, some of our cephalosporins, ertapenem, chloramphenicol, trimethoprim, tetracycline, and tigecycline. This extensive panel of intrinsic resistance is why susceptibility profiles for Pseudomonas that you get back from the lab oftentimes don't include very many drugs on them. They're simply intrinsically resistant to many of the compounds we have available to us. Intrinsic resistance is, of course, not limited to gram-negatives, and we have some really important examples in gram-positives. Maybe most impactfully are Enterococci. All Enterococcus species are resistant to all cephalosporins. You will never treat an Enterococcus successfully with a cephalosporin. It has an altered penicillin binding protein in its cell wall, which these drugs simply don't bind to. And I think it's also worth noting that some Enterococcus species, like Enterococcus gallinarum and Cassia flavus, are intrinsically vancomycin resistant. This is not VRE. This is just an Enterococcus gallinarum or Cassia flavus.